first thing first, I want to thank the organizers for the Congress and the ability to present and share with you our latest data on, what the heck, okay, on hypothalamus-driven alteration in ALS. So we started with this paper that was published not a long time ago that showed in, I'm not going to use that thing, in ALS, in sporadic and familial cases that these patients had a significantly lowered hypothalamus volume. But the most interesting finding in this study was that sporadic cases, well, the pre-symptomatic cases of ALS had also the hypothalamus being altered decades before the symptoms onset. And so for those who are not very familiar with this key brain structure, the hypothalamus is located in the middle of your brain, and it's the key structure to regulate pretty much anything from metabolism, reproduction, circadian rhythm, as well as sleep. And in this study, we are very interested in the lateral part of that structure, being the lateral part, the LHA, and is mostly involved in sleep regulation with the MCH and RX neuronal population. So we started off with a collaboration with the clinic in Ulm, Germany, and we had over 100 participants on this study. So we performed a couple of tests, had extremely stringent exclusion criteria, removing all participants that had respiratory defect or any comorbidities that would impact their sleep, and only ended up with 65 participants, 32 controls and 33 ALS patients. And so one of the most interesting findings here is that when asking them if they had sleep problems, all of them would tell us, like, no, we sleep perfectly fine. But when we analyze their sleep patterns with the hypnograms that you can see on the left part, they do exhibit clear sleep alteration by having more wake episodes and less NREM. So NREM is what we would like to is what we characterize as deep sleep. So that was interesting because they did not complain about any sleep, but in fact they had sleep alteration. But those are ALS patients, so maybe this alteration are mediated by the disease progression. So we had the chance to have a look at a second cohort of pre-symptomatic patients over 59, and we had the same exclusion criteria and exactly the same experiment done on them, and ended up with 19 controls and 28 gene carriers of ALS, and they also did not have any respiratory defects or comorbidities that would impact their sleep. And so, same thing, they do not report any sleep change compared to the controls. But when analyzing the hypnograms, also on the left, what you can clearly see is that SOD1 and C9 gene carriers have significantly more wake, and also for the C9s, less NREM, so less deep sleep, more wake in those patients, well, not in those patients, in those ALS gene carriers. So we, with that in mind, we wanted to know, well, can we translate this finding in our mouse models? And if we can we target and rescue that in our mouse model to then translate it back to the patients? So in the lab, we're using the SOD1G86R mouse model and fast delta NLS. And for those two, we're only interested in the pre-symptomatic stage of the disease, so the one in pink, and the rest is not really of interest for us for now. So the first thing we characterized in this mouse model was the hypothalamic alteration. So we started off with MCH, and in SOD1 and FAST, you can clearly see that they have less MCH positive cells, indicating that they are losing this neural population and that their hypothalamus is undergoing some alterations. But the counterpart of RX neurons remain completely unchanged in those two mouse models at pre-symptomatic stage, so there's just one being impacted there. So we went on and dwelled the physiological consequences of this hypothalamic rewiring, because one is impacted and not the other one, so we wanted to understand better what consequence would it have on our mouse model. And to do that, we recorded them with EEG implants over a 24-hour recording period, and we obtained those hypnograms shown here. And the one thing that you can see straight from those figures is these straight lines in SOD and FUS, which are not normal because, as you can see here on the white, they're not present. And in the white part, this is the non-active phase, so when the mouse is supposed to be sleeping, and the gray when the mouse is supposed to be active as well. And so when we quantify those, what we found is the same as in patients and pre-symptomatic gene carriers: increased wake episodes and less REM, which is the the dreamy stage where dreams happens, and the NREM, the deep sleep. So they have less sleep, more wake as well in these animals. And so, as I mentioned, MCH neuron is completely lost, so there's no point of targeting those, but orexin neurons are still present. So there is a possibility that we can reverse this phenotype by targeting the orexin neurons. And so to do that, we repurpose a drug that's already there on the market, at least in the US. And it's the suvorexant hypnotic sedative molecule that targets the 2 receptor of the orexin with an antagonist effect. And it's used in psychiatry and endocrinology to improve sleep, and it works pretty well by decreasing the wake and then therefore increasing the sleep phases. And so we gave this drug to our animals by doing the same experiment, EEG as well. What we found in SOD and first animals, I'm only showing you SOD results here due to the time limitations, but we were able to decrease the wake episodes of this mouse model back to the wild-type baseline levels, which was already quite nice to see. But when looking at their sleep, both the REM and NREM, we were able to increase that back to, again, the wild-type level. So that was a very nice finding of ours. And so to conclude, what I would just show you here is that we've had an hypothalamic atrophy and a wiring occurring decades before the symptoms onset in both ALS and ALS gene carriers and also our mouse models, and that those sleep alterations are present in these 2 cohorts, devoid of any respiratory defect, but with an increased wake, decreased NREM, so less deep sleep. They have an immense sleep fragmentation and difficulty to initiate their deep sleep as well, with much more defect. And all of these defects are recapitulated in our ALS mouse model at pre-symptomatic stage, so before any motor symptoms onset or else, with the hypothalamic atrophy, neuronal loss, the increased wake, and decreased REM and NREM. But because those alterations are orexin-mediated, we were able to completely rescue these alterations using repurposing the suvorexant in our mouse model. So that was nice to see we could accurately rescue these. And all those findings in terms kind of tell us that non-motor phenotype are actually very important in ALS as they occur before the symptomatic phase, and that they should be also taken into account in the pathology. So with that, I would like to thank our sponsor and our collaborator, Professor Ludolf and Dr. Lang in Universität, Dr. Lupi Lyon, Dr. Bobra as my supervisor, and Dr. Lupi, who's the head of the department here, and thank you for your attention.
Thank you. Any question?
Hello, thank you for your presentation. And so did you find some correlation between the functional score like ALSFRS and the NREM or the REM decrease that you find on patients?
So I did this correlation and there were nothing to be found there. It doesn't seem that the scoring, ALSFRS score, is correlated with sleep deficits, at least in all cohorts.
How specific are these alterations in human? Like, can we use this very early abnormalities in the hypnograms, like a biomarker of early changes in human? How much specific is that? Those increase in.
That's a good question.
Is there any other disease that can actually do a bias about those results?
So it seems that in other neurodegenerative diseases we have the same results, but they do complain of sleep alterations. Here they do not self-report any alteration, they do not complain about it. So that was a very striking findings of ours to see that they they would not complain about it, but the alteration are really present. So in sort of biomarkers, maybe we could look at more specific patterns, sleep patterns that are deep inside the EEG recording that we have. Those are indeed being currently used in Alzheimer's and Parkinson's disease as biomarkers. So those, yeah, clearly those can be used as biomarkers.
Thank you.
Thank you for
First
your
question?
Yeah. Thank you for your presentation. And I have a question pretty similar. What effect this sleep deprivation may have on the disease progression, according to you? Can we, for example, we know that in other neurodegenerative disease, there's a link between sleep abnormalities and the accumulation of some proteins, and it can be the same in ALS?
Yeah, definitely. If you have sleep alteration, it's most likely that you're going to have more oxidative stress. It will not renew your cell contents either. Which one appears first? That remains a question. But definitely, sleep alteration would not help at all with the disease progression. But that has to be assessed actually. That has not been looked at. Nobody is looking at sleep in ALS for now. So yeah, that's a good question.
Is it a quick one?
Well, we need to be very cautious on that, I think. It might be prodromal, like in PD, like in Alzheimer. It might be prodromal. You have sleep deprivation, you have sleep alteration when you're entering the disease. So it's the same for ALS. So let's try to develop this, to prove it, before entering in your, protein aggregation, sleep, it's very... So I think this, what you show is really great, and we need to work on this to see whether or not it could be a prodromal phase or, very early, and this could be very, very important. Also to tackle the patient very early. Because it could be, like a questionnaire to GP, and say whether or not you have sleep, problems, and you may enter, one of the neurodegenerative disease.
Yeah, I'm not saying it's specific to ALS, but when you see this alteration in gene carriers that are they were around 30 years old, it's definitely in the pre-symptomatic phase. So makes you wonder.